What I'm going to do is to give an overview on gene-environment interactions and risk of type 2 diabetes. We know that diabetes is a complex disorder. It's influenced by both genes and environmental factors. There is convincing evidence that type 2 diabetes has a strong genetic components because we know it is clustered in families and has higher concordance in monozygotic twins than in dizygotic twins, and also has higher prevalence in certain ethnic group. And in the past 2 or 3 years, more than 40 common variants, genetic variants, have been identified for type 2 diabetes through genome-wide association studies. But we also know that type 2 diabetes is heavily influenced by environmental factors. It has strong link to many dietary and lifestyle factors. Migration studies have shown that individuals, when they migrate from developing countries to obesogenic environment, and their risk of becoming obese and developing type 2 diabetes is increased substantially, even though there is no change in the genetic factors. And we also know that type 2 diabetes can be secondary to other conditions. So there is no question that type 2 diabetes are influenced by multiple genetic and environmental factors. So that's the reason we need to look at both the genes and the environment when we talk about predicting type 2 diabetes as well as prevention of type 2 diabetes. Epidemiological studies have been very successful in identifying dietary and lifestyle risk factors for diabetes. We know that body weight, obesity, is the single most important risk factor for type 2 diabetes. Independent of body weight, physical inactivity and smoking are also important determinants of type 2 diabetes. Many dietary factors have been shown to predict risk of type 2 diabetes independent of body weight, physical activity, and other lifestyle factors. For example, the type of fat and type of carbohydrate have been shown to play a major role in insulin resistance and diabetes. And also, our cohorts and many other studies have shown several foods and beverages may predict the risk of type 2 diabetes. For example, whole grain intake, high intake of cereal fiber, and higher consumption of coffee and dairy products are protective against type 2 diabetes. Moderate consumption of alcohol is also protective against type 2 diabetes. Several micronutrients have also been shown to affect insulin resistance and type 2 diabetes. These include chromium, calcium, iron, magnesium, and vitamin D. As I mentioned earlier, in the past several years, more than 40 common genetic variants have been identified for type 2 diabetes through GWAS. However, those genetic variants explain only a small proportion of familial clustering or heritability. So the question is, what are the sources of missing heritability for type 2 diabetes and other complex diseases? Those could be due to rare variants which haven't been identified yet, and it could be due to other structural variation, and it could be due to measurement error in phenotypes or heterogeneity in the phenotypes we are studying. Another possibility is that the heritability may be overestimated because the twin studies tend to overestimate the heritability coefficient. And finally, the sources of missing heritability could be due to gene-gene interactions or gene-environment interactions. So that's the reason we want to focus on the interactions between genetic and environmental factors. There are several very good rationales for study of gene-environment interactions, especially for type 2 diabetes. First, this kind of studies, especially subgroup analysis, may explain more of the variance in disease risk. Secondly, this kind of studies may help us better understand disease etiology and identify new drug or intervention targets. And thirdly, gene-environment analysis may help define susceptible population or high-risk groups and provide individualized prevention and treatment. To study gene-environment interactions, recently NIH has launched a major initiative called Genes, Environment and Health Initiative, GEI. And this initiative includes two components. One is called Exposure Biology Program. The purpose of this program is to develop technology and biomarkers for dietary intakes, physical activity, environmental exposures, psychosocial stress, and addictive substances. And in parallel, the genetics program of GEI, the purpose is to identify genetic variants through GWAS studies, sequencing analysis, and functional analysis. So the eventual goal of this initiative is to study the interactions between genetic and the environmental factors. So under the GEI program, we are funded to conduct comprehensive gene-environment interaction analysis using type 2 diabetes as an outcome. So in this project, we have already conducted GWAS analysis among about 3,000 incident diabetes cases and 3,000 controls in two large ongoing prospective cohort studies conducted at the Harvard School of Public Health and Brigham and Women's Hospital, the Nurses' Health Study and the Health Professional Follow-Up Study. In both studies, we have collected detailed dietary and lifestyle information during 20 to 30 years follow-up. So we are able to use prospectively collected dietary information and lifestyle information to examine the potential interactions between genetic factors and environmental factors for type 2 diabetes. So I want to briefly describe 3 examples of gene-environment interactions we have looked at in the past several years. The first example is the interaction between iron intake and HV genotype and the risk of type 2 diabetes. We know that type 2 diabetes is a common complication of hereditary homochromatosis, and in those patients, about 50 to 70% of those patients with hereditary homochromatosis have type 2 diabetes. So the question is whether higher iron overload predict risk of type 2 diabetes in apparently healthy individuals. So we conducted a last case control study to look at prospectively whether biomarker of iron overload, such as ferritin, predict the risk of type 2 diabetes. So in this analysis, we found a significant positive association between plasma ferritin concentration and the risk of type 2 diabetes, even after adjusting for BMI, inflammatory cytokines such as CRP and the interleukin-6. In the Nurses' Health Study, we also found a positive association between higher consumption of human iron and the risk of type 2 diabetes. Human iron comes from animal products, is more readily absorbable compared to non-human iron from plant-based food. So this analysis was adjusted for a wide range of dietary and lifestyle factors for type 2 diabetes. So the next question is whether the association between dietary iron intake and the risk of type 2 diabetes is modified by HV variants. The reason we look at this interaction is because HV variants such as C282Y, H63D have been shown to predict higher iron overload, and also individuals with those genetic mutations or polymorphisms have higher iron absorption than those who don't carry those genetic variants. So in this analysis, we look at association between human iron intake and the risk of type 2 diabetes stratified by HV genotypes. So as you can see that among those with wild genotypes, there is no association between dietary human iron intake and the risk of type 2 diabetes. However, among carriers of H63D or C282Y, there is a significant increased risk of type 2 diabetes with higher consumption of human iron. So this suggests that those participants are more susceptible to detrimental effects of higher iron intake on risk of type 2 diabetes. So in summary, from this study, we found that the genetic variability of HV gene, H63D and C282Y, was significantly associated with increased body iron stores, which reflected by higher plasma ferritin levels. And also, in this analysis, we found carriers of H63D and the compound heterozygous tend to have moderately increased risk of type 2 diabetes, whereas the carriers of C282Y did not have significantly increased risk of diabetes. However, HV genotype appeared to modify the relationship between dietary iron intake and risk of type 2 diabetes. So the second example I'm going to describe is the interaction between TCF7L2 and the dietary carbohydrate intake in relation to type 2 diabetes. As you know, TCF7L2 is the strongest gene for type 2 diabetes identified so far. And the risk allele of TCF7L2 has been associated with impaired insulin secretion, decreased beta cell function, and also impaired response to agents promoting insulin secretion. So we hypothesize that dietary factors that increase insulin demand might enhance the risk of type 2 diabetes associated with TCF7L2 variants. One common matrix which has been used to characterize the quality of carbohydrates is glycemic index. So typically foods or beverages which are readily absorbable, such as refined starch and the sugar-sweetened beverages, have higher glycemic index. And the glycemic node is the cross product of glycemic index of the food and the total amount of carbohydrate in the food. So glycemic node is, it reflects both quality and the quantity of carbohydrates. In our study and several other large epidemiological studies, higher glycemic node diet has been associated with significantly increased risk of type 2 diabetes. So in this analysis, we wanted to examine whether higher glycemic load, which increased insulin demand, modified the association between the TCF7L2 risk allele and the risk of type 2 diabetes. In this analysis, we look at the association between the TCF7L2 genotype and the risk of type 2 diabetes stratified by different levels of glycemic load. As you can see that the association between the genotype and the risk of type 2 diabetes varies substantially across different levels of glycemic load. The association is strongest among those who consume the highest glycemic load diet, and the association was lowest among those who consume a low glycemic load diet, and the association was intermediate among those with an intermediate glycemic load diet. So this analysis suggests that dietary glycemic load modified the association between TCF7L2 genotype and the risk of type 2 diabetes. So in summary, this study suggests that carbohydrate quality of the diet modified the risk of type 2 diabetes associated with TCF7L2 genetic variants. And this suggests that the impact of carrying a TCF7L2 variant is magnified under conditions of increased insulin demand. And these findings may have implications for prevention of type 2 diabetes in individuals harboring TCF7L2 risk variants through dietary interventions. So the third example I'm going to describe is the interaction between dietary patterns and interactions between dietary patterns and a genetic risk score for type 2 diabetes. So So in our study and many other studies, dietary patterns are typically derived through factor analysis or principal component analysis using data from food frequency questionnaires. In our study, we derived two major dietary patterns. The Western dietary pattern was characterized by higher intake of red meat, processed meats, refined grains, high-fat dairy products, and high sugary drinks. Whereas the prudent dietary pattern was characterized by high consumption of fruits and vegetables, whole grains, low-fat dairy products, and higher consumption of fish. For genetic risk score, we summed the number of risk alleles for about 10 genes, diabetes genes, which have been identified through the genome-wide association analysis. So we calculated the genetic risk score for each individual. As you can see here, the distribution of the genetic risk score shift to the right for the cases compared to the controls, but there is a huge overlap between the cases and the controls in terms of the genetic risk score distribution. Similar to other studies, the genetic risk score is strongly associated with increased risk of type 2 diabetes. As you can see that there is a dose-response relationship between the number of risk alleles and the increase in risk of type 2 diabetes. The relative risk is about fourfold comparing those who carry more than 14 risk alleles with those who carry less than 7 risk alleles. However, the genetic risk score did not add to the prediction of type 2 diabetes beyond traditional risk factors such as BMI, physical activity, and dietary factors. So to look at the interactions between dietary pattern and the genetic risk score on risk of type 2 diabetes, we classified the participants into 3 groups based on their genetic risk score. No genetic risk score, medium score, and high score. So in this stratified analysis, we look at the relationship between Western dietary pattern score and the risk of type 2 diabetes stratified by the genetic risk score. As you can see that there is no association between the Western dietary pattern among those who have the lowest genetic risk score, whereas among those who have the highest genetic risk score, there is a strong, significant positive association between increasing the Western dietary pattern score and increasing risk of type 2 diabetes. And we also look at interaction between the prudent dietary pattern score and the overall genetic risk score for type 2 diabetes. However, we didn't find a significant interaction between this dietary pattern and the genetic risk score. And then we look at the major components of the Western dietary patterns and the genetic risk score in relation to type 2 diabetes. So for red meat, overall is significantly associated with increased risk of type 2 diabetes. However, the association was more pronounced among those who have higher genetic risk score, and the association was not significant among those who have lower genetic risk score. So it seems that those who have greater genetic susceptibility to type 2 diabetes are more susceptible to detrimental effects of red meat on risk of type 2 diabetes. We found similar interaction for human iron intake and the genetic risk score on risk of type 2 diabetes. Again, the positive association was more evident among those who have greater genetic predisposition to type 2 diabetes, and the association was not significant among those who have the lowest genetic risk score. So this slide shows the combined effects of the genetic risk score and the Western dietary pattern. So as you can see that the association between the Western dietary pattern and the risk of type 2 diabetes was more pronounced among those who have the highest genetic risk score. And this means that those participants who have greater genetic susceptibility to type 2 diabetes, perhaps some more responsive to the Western diet. On the other hand, you can also say that the association between the genetic risk score and the risk of type 2 diabetes was most pronounced among those who have the highest Western dietary pattern, and there is no association between the genetic risk score and the risk of type 2 diabetes if they don't consume a Western diet. So this suggests that consuming a healthy diet may counteract or attenuate the detrimental effects of the high genetic risk score on risk of type 2 diabetes. So a similar interaction has been found for obesity. This is a study Dr. Luce presented this afternoon. In this study, they found that higher physical activity attenuates the genetic predisposition to obesity in the EPIC cohort. So those analyses suggest that a healthy diet and a lifestyle may diminish or even abolish the detrimental effects of genetic predisposition on obesity and type 2 diabetes. And we know that both conceptually and methodologically examining gene-environment interactions is much a bigger challenge than looking at the main effects of the genes or the main effects of the environment. A major challenge is the sample size. Typically, you need much larger sample size to detect gene-environment interactions than detecting the main effects of the genes or environment. And the statistical modeling of gene-environment interactions is also complicated because you can model additive interactions or multiplicity interactions. And the study design for gene-environment interactions ideally should be prospective cohort studies. It cannot be cross-sectional or retrospective case control studies because you want the dietary lifestyle information collected before the diagnosis of the disease so that you can avoid recall bias. Another major challenge is that environmental factors, especially diet and physical activity, are measured with substantial measurement errors. So in this kind of situation, you can get biased estimates of gene-environment interactions. You can also have reduced power for gene-environment interaction analysis. And also, replication of gene-environment interactions is much harder compared to replications of the main effects of the genes or environmental factors. And statistically, the major problem for gene-environment interaction is the multiple testing, which is exaggerated when you look at many genes, many environmental factors, and you can have, especially in GWAS, you can have millions of SNPs to look at. You can also look at many different genetic models, and also there are numerous environmental factors that you can look at, not only diet, physical activity, but also other environmental factors. So in this kind of situation, the multiple comparison problem, I think, is going to be much, much worse than the studies looking at only the main effects of the gene or environmental factors. At this point, there is no optimal solution to the multiple comparison problem in gene-environment interaction studies. Similar to the main effects of the gene using GWAS, we have to rely on multiple large studies for replications, and also we have to look at the strength of the association, the biological plausibility, and the consistency of findings between experimental studies and epidemiological studies. So in light of the difficulties, methodological difficulties with gene-environment interactions, as I mentioned earlier, NIH has launched the Gene-Environment Initiative, and under the Gene-Environment Initiative, it has founded the consortium called Geneva. It's basically a study of both genetic and environmental factors for various disease endpoints. And this is the first wave of GWAS studies which was founded by the Geneva Consortium. And last year, this consortium, this initiative, founded another 8 or 9 large cohort studies. And in all those cohort studies, dietary and lifestyle information has been collected. So using Geneva, it's possible for us to not only look at the main effects of the genetic variants for complex diseases, but more importantly, to identify variations in gene-trait interactions related to environmental exposures, especially diet, physical activity, and other environmental factors. A major goal of this Geneva Consortium is to ensure the rapid sharing of data, both genetic data and phenotypic data, to the scientific community. So right now, all the studies that have finished the GWAS, the data, both the GWAS data and the environmental data, have already been deposited in the dbGaP. So coming back to the premise of gene-environment interactions, I think this kind of analysis have great potential to explain more of the variance in disease risk, and also has potential for us to better understand disease biology and perhaps identify new drug targets. But at this point, the evidence is not sufficient enough for us to make individualized nutritional or dietary recommendations. This may change in coming years or perhaps decades when we have more and more evidence, solid evidence for gene-diet interactions. So I want to stop here and thank you very much for your attention.